We have two major goals when we do immunogenetic research in CLL. One is to always try to identify novel biomarkers for assessing disease prognosis and prediction. And the other is to try to advance the existing classification schemes for CLL patients. Of course, this has proven a very fruitful field. And as I told you, the somatic representation analysis is now considered as a one of the breakthrough biomarkers in CLL. But of course, also because in the latest years we are focusing on the study of B-cell receptor stereotypy in CLL. And there we have found specific subgroups of patients carrying similar immunoglobulin molecules. And these patients, when we study them at very different levels, so the genetic, epigenetic, functional, cellular, what we found is that indeed these patients represent very unique variants of the disease. So we have reached a level where we have partially at least overcome the extreme complexity of CLL, and we have found very specific disease variants, both at the biological and clinical level. And for example, in a study that we did last year, we have shown that at least a stereotype subset of CLL patients, stereotype subset number 2, as we call it, and it's one of the the largest subsets in CLL. And this subset is very interesting in terms of clinical prognosis because it represents almost 7% of patients with CLL requiring treatment. It has proven to have a very aggressive, to correlate with a very aggressive disease. And what we've shown in our paper is that patients belonging to this group do not benefit from the application of chemoimmunotherapy. And this is something really important because clinicians should always keep in mind and address this issue.